Thank you, Wes, for the introduction, and thank you very much to the organizers for inviting me to be here today. I'm sorry to disappoint those of you who saw an Australian on the program and were hoping that I would have a cool accent, but maybe you'll enjoy that later in the program. So today I'm going to talk to you about enabling clinical translation of high-throughput functional assay data. And so what I mean by high-throughput functional assay data is this class of experimental techniques that we call MAVES, or multiplex assay of variant effects. They come in two major types. There's DMS, or deep mutational scanning, which is focused primarily on protein coding variants. And then there's MPRAs, or massively parallel reporter assays, which are focused on non-coding variants. Now, my expertise is really in the deep mutational scanning area, although many of the things that I'm going to talk about today are equally applicable to MPRAs. And one of the things that I think is interesting about looking at coding variants is that we know that advances in genomics really start with the gene. So if we think about what's our relative level of understanding of some of these types of variation, coding variants, I think we understand the best. We've understood what the grammar is since 1961. Non-coding variation, I think that through some large-scale efforts, through projects like ENCODE and IGVF and the contribution of many other investigators, we're still trying to get a handle on what non-coding variants do. And then structural variation, while there are some very well-characterized examples, I think as a class, we're still really trying to get a sense of it, and I'm not sure that it really makes sense to think about what is a grammar for structural variation. And the consequence of this is that on the one end, we're still very much cataloging all of the variation. But on the other end, because we have this better understanding, we can start thinking about what we can do to translate the data and to make an impact clinically. And I think that this is a really good audience to be engaging with about the possible translation of coding variants, because I know that in the cancer space, that is a lot of what you all are working with. And so the problem that we're trying to solve is in a very broad scale, is this problem of variants of uncertain significance. So this is just a plot showing missense variants in ClinVar over time. And so on the right here, so when we first made this plot, I think in 2020, we thought that the trend was likely to continue, and it appears that the trend has in fact continued, and we now have many, many thousands of VUS that are missense variants. And again, this is the type of variation that we have the most tools to be able to understand and interpret. And then the donut plot is just showing you the total in ClinVar as of earlier this year, and we can see that probably more than 80% are VUS. So how are we going to solve this problem? Obviously, we're not going to sequence our way out of this, and so I'm going to tell you today why I think that high-throughput functional data can really help us solve this problem of variants of uncertain significance. So if we think about these three key challenges, we have data generation, we have data sharing and standardization, and translational data models. And the rest of this figure, you sort of representing the researcher, databases, both international and regional, the role of the variant curator, the role of the patient-facing clinician who is returning the results to the population, and then we hope that the patient need will feed back into motivating additional research studies. And so we'd like to really fulfill all the kind of pieces of this cycle and kind of close this loop. So I'll start by telling you about data generation. I am assuming that many of you are familiar with the existence of these techniques, but maybe not so much with the mechanics of them. And I think that it would probably be useful to talk a little bit about how these experiments are being done. And I'll give you a couple of examples from studies that I've been involved in that I think are particularly relevant for cancer. So in one of these assays, we start by creating a library of genetic variants. And in the deep mutational scanning context, these are going to be variants of a protein coding gene. So we start from a target sequence. We've got our nucleotide sequence, we've got our codons. We can translate it to figure out what the amino acids are, and then we can introduce variants into this target sequence, and here we have a missense variant. But we don't want to just make one variant and test it. We don't want to make 10 variants and test it. We want to make many thousands or even tens of thousands of variants of a gene encoding all possible single amino acid changes in that sequence. And so we can do multiple single nucleotide changes. We can do full codon swaps. We can do in-frame deletions of a single codon. And then we can make this diverse diverse library of variants, and then transform it into cells to create a population of cells where each cell has a single variant, and then we can measure what that variant does in terms of how the cell performs in the assay. So there's a lot of different ways to do this. We can use cDNA-based library synthesis, which we can either do ourselves using mutagenic PCR. You can order these from a vendor like Twist. There's a variety of CRISPR-based methods like saturation genome editing, where we can introduce CRISPR guides to make a double-strand break and then use a provided repair template to incorporate specific mutations that we've programmed in. And then there's the couple of emerging areas that I think are worth mentioning, which are CRISPR base editing and also prime editing, CRISPR prime editing. And while these are not mutational scans or in the sense that they don't do things at a saturation level, I think that bioinformatically and sort of practically speaking, they're very similar. And I think that because these techniques tend to be less expensive and less technically challenging than doing a full mutational scan, and also are amenable to exploring a lot of different phenotypes and also are easier to work with in primary cells, we will start to see a lot of data using these types of techniques in the near future. And I think that everything that I'm going to say today about the analysis, the handling, the clinical translation, et cetera, these are all things that apply to these types of data sets as well. And we're very excited to incorporate these kinds of data sets as they become more abundant. So once we have our library of genetic variants, we want to figure out what they do so we can perform a functional assay. One of the simplest examples, and one that is particularly topical for looking at cancer genes, is to examine cell growth. So we can take a sample of the population and see at what frequency do all of our variants appear. We let the cells grow for a while. We take another sample of the population, and then we see what the frequency is afterwards, and then we can compute essentially a functional score, which is essentially a measurement of the enrichment or depletion of each of these variants. So this is using high-throughput sequencing essentially as a digital counting instrument to profile how many copies of the variant do we see in the population before and after we do the assay. And so we often visualize these functional scores in something we call a variant effect map. So it's this heat map structure where in this version, the positions are the columns. These are the positions in the protein. And then the rows are the amino acid that is being mutated to. Traditionally, a loss of function variant is depicted in blue, and a potentially hyperactive variant is depicted in red. And if you think that seems backwards, ask me about it, because I had a bunch of arguments about why this should be the standard. But you can see that there's a lot of structure in these data sets, right? There are certain positions that are intolerant to mutation or seemingly intolerant to mutation. There are others that are very permissive, and then there are some exceptions that seem like they may be interesting and worth following up. But of course, we can do a lot more things. There's a lot more functional assays than just cell growth. So some of the other techniques that have been used for mutational scanning, we can look at molecular properties. So this includes phenotypes that can be detected using confocal microscopy, which is obviously a lot of different things. Cell surface expression, other stuff like that. We can do anything with a fluorescent reporter. Now we can, and then there's also an emerging area of using single cell readouts. So you can imagine that if you wanted to look at a transcription factor, getting a bunch of single cell transcriptomes for each variant would give you a lot of information. I think that right now this is very, very expensive, very, very technically challenging. But if we've learned anything from the history of genomics, we know that this will become much easier and cheaper to do over time. So with that, I'm just going to give you a quick example of a couple of different studies that are maybe a little bit different from like a traditional mutational scan that I've been involved in that I think would be particularly relevant for this community. So the first one is using mutational scanning to understand cancer drivers. So here, this is a mutational scan of the transmembrane domain of MPL, the TPO receptor, and I'm not going to try to explain to you what this does, because I think that probably at least half the people in this room understand the biology of this system much better than I do. But the takeaway here is that we've done a mutational scan of the transmembrane domain. So if we look at what that heat map looks like, so this one's inverted, or not inverted, it's rotated. So here we have the positions are the rows and the amino acids are the columns. I apologize for that, but it's kind of a nice one for a slide, right, because it's really short. And we can map it onto the helix. And so one of the things that I hope you can take away from this is that if you look at what's in the heat map, and again, the sort of potentially activating mutations are in red or this peach color, there's a periodicity here, and if we map it on the helix, there's a single face of the helix that seems to be the important part of it. I think it was known that this helix was interacting in the membrane, but it was not necessarily known which face of the helix was important or that it was a whole face like this. And so that was kind of a neat mechanistic insight that we could provide by doing this experiment. Now, what I think is really cool about this example is that there's this particular variant here which you see pops up, bright red in that mutational scan, which is this S505N, which is a very well-known driver mutation that was already well established. And so what we did was another mutational scan, so a very similar experiment, but in this time, instead of using the wild type or the reference background, we use the driver as the background. So this is essentially everything here is in relation to what this S505N driver is doing. And you can see that there are all of these mutations that pop up as bright red, which only appear to be doing something in the presence of the driver mutation. And they appear to be totally neutral or even slightly deleterious in the absence of the driver mutation. And if we look in the literature, we did a collaboration with a group in Italy who had a bunch of patient data on this. We can see that there's a bunch of these that have been observed in patients, and they all pop up in this mutational scanning data, and as well as many other ones. And so I think this is the kind of insight that we can get from some of the functional data that may be very important and relevant clinically, and it's also kind of a different dimension of exploration if you're thinking about kind of research opportunities or ways to apply this technology. And there's also a triple mutant there, which I didn't mark on the slide, but both of those are in the heat map as coming up as more active than just the driver. I also think this is an interesting example of how oftentimes these, the multi-mutants it's not additive. These are not like a linear additive combination. There is some sort of, epistatic interaction, if you will permit me to use epistasis in this way. Some people don't like that, but it is a cool example of what you can learn from functional data. The other example is I'm going to show you a little bit of brand new unpublished data using deep mutational scanning for pharmacogenomics. So this is looking at PARP1 and PARP inhibitor resistance. So we can take a PARP1 variant library, transfect it into BRCA1 null background, and select for growth with a panel of different PARP inhibitors. And the intention here is to be able to inform patient treatment based on genotype. So we can look, for example, at the PARP inhibitor rucaparib. There's this one variant that is the strongest variant for rucaparib resistance in our study. And you can see it's this tyrosine down here that appears to be interacting with kind of the tail of rucaparib. And this pops up as being a drug-specific variant. Another example is one for olaparib, which does not, that previous variant doesn't pop up for olaparib, that is like right down here where the PARP1 catalytic domain is interfacing with HPF1, and that's kind of where this drug is interacting. So we can see that and this is a this animation that's looping is from molecular dynamics simulation, showing what that variant might be doing. And it does appear to be another drug-resistant or drug-specific interaction that's mediating resistance. And so I think kind of the takeaway from this is that here are a couple of examples where we were able to understand what the mechanism is, or we already knew what the mechanism was, but that if you were to do one of these studies, there would be a lot of other things that would pop up as being drug-specific that you might not know what the mechanism is, and that would provide a lot of insight, whether you're interested in response to therapy or drug development or anything else like that. So we think that this is another very interesting opportunity for future work, particularly in the cancer space, by looking at resistance and ways to try to at least extend the time before resistance develops. So I hope that so I'll add just on the previous slide that because this is done in a living cell with no modifications to the protein, it is often really a lot better to, it's a much better way to look at some of these interactions. Okay, so I think I hope I've shown you that heterogeneity is both a great strength of this field. It is also a great challenge that we have to confront. We have a lot of different assay systems. We have a lot of different assay types. We have different ways of making libraries. We have different levels of coverage. We have all of this kind of complexity that we need to contend with. And so one of the really important things that was a challenge that we as the field needed to face was what were we going to do about data standardization and sharing? Because and to do that, so we started this project to build MaveDB, which is now the community database of record for multiplex functional data. Before MaveDB, kind of every single one of these papers had its own supplemental table that had some number of variants in some format. I have personally curated probably almost certainly more than 100 papers into MaveDB, and I have never been able to use the same analysis notebook twice. Like every single one has been unique, even ones from the same lab that were published at the same time. I think that those of you who have done a lot of work with the literature and doing some of this bioinformatics will know exactly what I'm talking about. Everybody just does everything a little bit differently. But that's a huge impediment if what we want to do is allow people to work with this data at scale and use it for other things. So we really want to standardize it. I think this has been very successful. We now have a lot of people who are depositing their data to us directly at publication time or pre-publication. We now have almost 2,000 data sets and 7 million variant effect measurements. So here's just a plot of the growth over time. So the green line is the number of data sets, which is defined as an assay that was done on a target. And then the blue bars are the cumulative number of variants. And so this 2024 bar is as of, I think, the end of May this year. So we're definitely not leveling off. There's still a lot of variants that we're hoping to capture. The data is diverse, but it's mostly human. So MaveDB, the primary sort of goal of MaveDB is to be a data warehouse for the community. So this dark blue in the bottom here, which is about half of it, those are the human targets. There's this light blue, which is almost a quarter, which is artificial sequences. This is actually a very popular technique for people who are doing things like de novo protein design, because it allows you to do an unbiased search of sequence function space around your computationally derived target, and that can be really valuable if you want to do engineering stuff. And then there's a bunch of other proteins from various model and non-model organisms. And we also have a variety of different genes that I think are particularly relevant. So these are genes on the ACMG secondary findings list, and of course many important cancer genes, some of which we actually have multiple data sets for. So that's multiple different functional assays have been performed on these targets. We have all of that data. It's in the same format. So if you want to look at, where the assays agree, where the assays disagree, and try to understand, what might be going on there, at least, we're now one step further in terms of enabling people to do that sort of work. The community has really rallied around MaveDB in a very encouraging and satisfying way. It's a very open community. We're very committed to data sharing. And we also have a very complementary project, which is run by Fritz Roth, who's now at Pitt, which is the Mave registry, which tracks data generation efforts. So people who want to do a multiplex assay can register their project on Mave registry. People who maybe If you have seeing a bunch of variants in a particular gene and you would like somebody to do a multiplex assay as part of a collaboration, you could also register a project on MAVE Registry and say, hey, I really need someone to do an assay on this gene and develop a collaboration. And there's been a lot of really successful international collaborations that have been nucleated by this, because there are so many genes for us to scan, it really doesn't make sense for people to be accidentally competing with each other. And so I would encourage you to check this out, even if you don't want to do one of these experiments. But if you have that need, a lot of times there are technology-based labs who would be happy to take on something where there's a clear clinical application. So in MaveDB, we've built a website. It's got a bunch of stuff in it. We're automatically generating data visualizations. We've got some link outs to other resources, including the UCSG Genome Browser. We collect various text data, like an abstract and methods. We give you details about the experimental target and a preview of the data. And so we're really trying to build this out so that it's ever more attractive for researchers who are generating these data sets to upload and share their data. We would like it to become something where people see that there's value in uploading the data when they're preparing their preprint, and then they're already ready to make it public once their preprint or publication comes out, because we're providing them with like some visualizations and some analysis and some other stuff like that, rather than it being one more thing that the beleaguered graduate student has to do before they can press submit, because that's how these things can very easily get dropped from that list. We are also trying to make these data sets easier to deal with by downstream consumers of the data. So in particular, other websites, potentially clinical dashboards and things like that. So to do this, we've developed minimum information standards and a controlled vocabulary, because as anyone who has tried to deal with this kind of experimental data at scale knows, if someone writes you a beautiful abstract in plain text, that is not useful. If what you're trying to do is classify and categorize the data, you would much rather have them pick from a list of 10 or 15 controlled vocabulary terms and have that be presented in a structured, standardized format. And so that is something that we've now developed through Atlas of Variant Effects in collaboration with Alex Wagner and others. And we have implemented this, and it captures kind of all of these steps that I told you about. An example, we have a project summary with like the title of the paper and what publication it is, a summary of the experiment. We can include OMIM and Mondo codes for like what the disease relevance the experimenters are assigning to it. We have our own controlled vocabulary for things like library generation and the phenotypic assay, which are more specific to our field. And we're now working to get these kinds of terms adopted by sort of more mainstream ontologies to, again, promote the kind of our capacity to share the data and integrate it across a wide variety of different resources and knowledge bases. Another thing that we'd really like to do is to, again, use the standards to promote data deposition. It needs to be, carrots and not just sticks if we're going to get people to share their data. So for example, if someone deposits their data, then we could, and they fill out a little web form that tells us what goes in the minimum information standards, we could maybe generate them a cool PDF report with a gold star on it, and then they can submit that as a supplemental data with their paper, and then people will know to look for that as sort of a mark that the data has been shared and it's available, and so on. And so we're really trying to think creatively about how we can encourage people to participate in data sharing and make it worthwhile for them. One of the big challenges that we had to face from going from the original kind of vision for MaveDB, which was very much like a data warehouse that was intended for researchers, and so researchers could upload and download whole data sets, we weren't really at that point thinking about individual variants, and we certainly weren't aware of the level of interest from the clinical community in terms of using these data sets for clinical variant interpretation. That was not something that we were thinking about when we started. And so we ended up in this very challenging situation where we had had curated all of this data that we could not really map to the human genome. So in collaboration with Alex Wagner and Jeremy Arbesfeld in his lab, who was here at the meeting, we undertook this process of mapping MAVDB variants to the human genome. And we used this GA4GH variant representation specification to essentially allow us to represent a single variant object that's with respect to whatever experimental sequence was used in the assay, which may not and often is not reference identical, and then whatever that is on the human genome reference. And that allowed us to maintain the sort of continuity and the provenance. So for people who are very concerned about making sure that what was actually tested in the assay is consistent with what's important for them when they're looking at a patient, we could keep all of that together. And so it's kind of these composed objects. It's based on JSON schema, which is not anything that you would want to read, but it's really, really good for sharing information between computer systems, which is really the goal here. Because the goal behind all of the standards work is to enable data sharing and integration. So here's just an example. Here's just a slide showing some of the places that we're sending data to. So, UniProt, the data is in Ensemble VEP. People who were at the bioinformatics workshop heard about the G2P portal from the Broad Institute here. You can see the data as a track up in the UCSC genome browser, et cetera. And then we're also generating a lot of data as part of the IGVF consortium, which is a large NHGRI consortium focused around understanding the function of variants. So that's good for researchers, and we want to make sure that we can share all of the data. But to actually build real clinical data models, we need to do more than just say, here's a variant and it has a score. And so I'll talk to you a little bit now about how we are using multiplex assays as functional evidence, how we can think about it in that context. And what we're doing to try to put together clinical data models that are more suitable for things like curation dashboards and so on. So if we want to use MAVs as functional evidence for variants, usually I present this with the table from the 2015 Richards guidelines about germline variants, but I figured since I am here, I would use this very nice table from these more recent guidelines on oncogenicity. And what was nice for me is that the box that we're concerned about, these ones that are highlighted in red, which say well-established functional studies show no oncogenic effects, and well-established functional studies supportive of an oncogenic effect, that language is basically exactly the same in Richards, although Richards obviously doesn't say oncogenic. It says pathogenicity. So that's what we want to do. We want to show that our data is a well-established functional study. We want to be able to tell you whether it does or does not have an effect. And then we want to make sure that we're explaining to people in a way that they feel comfortable with, what's going on there. And I will add that, in this rubric, right, that it's either 4 points for or against. So that's enough to get you to likely benign or like a pretty hot VUS with just the functional data. And so it's pretty likely so that's, again, the importance of making sure that people can use this data, because it's going to be relative, it gives you a lot of points towards actually getting an answer that's going to help the patient. So when we look at MAVE variants, we can plot, like, again, the distributional functional score. So here's some example from the saturation genome editing of BRCA1, which was published in 2018, which is kind of remains one of our sort of exemplar data sets when we're talking about this. So here we're just looking at, and these are it was all single nucleotide variants. This was done by editing the endogenous locus and HAP1 cells. It turns out that haploid cells really don't like it when you break the DNA repair pathway. So it's a really excellent model system for looking at these kinds of genes. And so the bars are colored by what the effect is. So you can see we have a distribution centered over like -2 that's red, that's the nonsense variants. and also the canonical splice variance. So that's our sort of total loss of function distribution. And then we have the dark blue synonymous distribution, which is around 0. And so those are the variants that have like very little effect. So this sort of bimodal distribution is pretty typical of these cell-based assays. Cells are pretty good at buffering intermediate effects, so things tend to get pushed to one extreme or the other. So if we want to think about evaluating the strength, we can, for example, look at variance in ClinVar. So now on the top here, I'm showing you the data from that BRCA1 study. On the second row, I'm showing you data from PTEN, which looks a little bit different. So if we look in ClinVar at the distribution of pathogenic and benign variants, so these are variants that were all classified before the multiplex assay data was made public, so there's no risk of circularity. We can see that there is a very, very clear separation of pathogenic and benign variants. All the pathogenic ones have a negative score. The benign ones have a score that's near 0. There are a few that don't agree. It turns out that some of those are actually wrong in ClinVar and that the assay was right, and there's reasons for that I'm happy to talk to you about if you're really curious. And then the other thing that we can use to kind of try to calibrate this is we can use this distribution of nonsense and synonymous variants, and then we can kind of draw these vertical line cutoffs, and we can say, okay, if your variant looks like a nonsense variant, it's probably damaging, and if it looks like a synonymous variant, then it probably isn't. So the BRCA1 case on the top there, that's kind of our ideal case. And then you have other cases like PTEN where, obviously people have been studying PTEN for a long time, but there were only 2 likely benign variants in ClinVar for PTEN. Makes it very, very difficult to use this as a basis for classification. This, I think, is a more typical case where you may have a handful of variants, but they're all pathogenic variants. So if you run a clinical lab, please deposit your benign variants in ClinVar. We really need to see them. But we can do something here. This assay is maybe a little bit messier, but we can still do this nonsense synonymous split. And there's guidelines from the ClinGen Sequence Variant Interpretation Working Group, the Burnich guidelines, that tell us how to calculate an odds ratio that establishes the strength of evidence within the Richards guidelines in order for us to tell whether something is strong or supporting or so on. And so if we use that, we can reclassify a lot of VUS using MAVE data. So here's an example of a study that we did in collaboration with Ambry Genetics, where they provided a bunch of information about patient variants that they had seen, many of which we wanted to reclassify. And so for BRCA1, we were able to reclassify about half of the variants of uncertain significance. Most of them ended up being likely benign. P53, even more of the variants were reclassified. P10, not so many. We were able to move a few from VUS to likely pathogenic, but I think this is still, especially this is a very early example of using these data sets to try to reannotate or reclassify real variants, and I hope it shows you that the sort of the power of doing this and the kind of the importance of making sure that we can do this at scale. So we have now a feature in MaveDB that's in sort of preview form, and if people are interested in it, I would love to talk to you during the conference. So where we can expose the Mave variant information on individual variants. Again, this is something that we were not thinking about at all when we started the project. We've now gotten to the point where we can generate a page for each variant. We can tell you based on some of these calculations that we can do. So here at the bottom, you can see the odds path, the odds of pathogenicity, and then whether it's PS3 strong or supporting or moderate and so on. We can show you the information about the individual variant, where it falls in the distribution, and for this one we can tell you that it's functionally abnormal. And so this is the kind of thing that we're hoping to have in our website, so that for people to do a deep dive, but also that we want to make sure that we are exporting widely and exporting in a format where people can incorporate it into their own platform, so that they don't necessarily have to come and visit us. We can provide structured data. And so to do that, we're developing a new standard that we hope will be adopted broadly for functional data. It's what kind of combines the work that we've done on the minimum information standards and controlled vocabulary that I told you about before with existing work from the ClinGen Sequence Variant Interpretation Working Group. And we're doing this through the Global Alliance, which is a really fantastic organization that has a lot of, the process for developing these standards and supporting implementers and so on. So we're really excited about this work, and we're putting together a draft standard now. So if you are interested in being an early adopter, also please come and talk to me. We would love to hear about your concerns. And again, most of the people who I interact with on a day-to-day basis are much more focused on germline variants and pathogenicity. And I know that there are a lot of very important differences when you're looking at oncogenicity. So for people in this community, I would really like to make sure that we're developing something that can work for everybody, because I think that obviously from the perspective of generating the functional data, we think you can use it anywhere that you want, but we want to make sure that it's packaged in a way that's compatible with your workflows. So this is going to enable a lot more discoverability and translation. So the data is actually already in Decipher in a sort of a limited form. We're putting it in the ClinGen Link Data Hub. We've got plans to expose a lot of the MAVEDB data in ClinVar. Within Australia, we're going to be putting the data into the Sharient platform, which I think we'll hear about later this conference. And then also New South Wales Health Pathology, which is one of the largest clinical testing labs, probably the largest clinical testing lab in Australia, is going to be putting this into practice. And so really, we're going to hopefully get a lot of really good insights into how to present the data. So if you would like to get involved in this, again, yeah, please, I would love to hear from you. Let's see. So I was really excited to see that tomorrow there's an entire session about equity in genomic medicine, and so I thought that I would tell you a little bit about one of our contributions in this area. So one of the things that we always aspired to in the multiplex assay community was that because we're looking at every variant, not just variants that have been observed in people who have been sequenced, which is primarily people from European-like ancestries, we were thinking that these data sets in particular would be potentially very useful for reclassifying variants for people from non-European-like ancestry. So in this table, I'm showing you the number of variants of uncertain significance in BRCA1, P53, and P10, which are the ones that I showed you in that previous study where we reclassified a bunch of variants. So if you don't apply functional data, then there's this highly statistically significant difference where people of non-European-like ancestry have many more variants of uncertain significance than people of European-like ancestry. But if you have saturation mutagenesis data, then that difference becomes no longer significant, because it is working against a lot of these ascertainment-based issues that are driving, in some sense, driving this inequity. So we can reclassify many more. Another way to look at this is we are reclassifying many more variants successfully in non-European-like individuals. And if we look at variants of uncertain significance in GenCC, which is one really nice list of all of the genes that are potentially clinically relevant, we can see that before multiplex assays, if we had a really good multiplex assay for all of the genes, we would again be able to reclassify a very large proportion of genes and get rid of this help get rid of this population-based disparity. Now, if we're going to do this, we need to actually try to educate clinicians about multiplex assays. I can't just give clinicians the data and say, here's the data, it's good, trust me, use it for your patients. We want to make sure that people who are applying this stuff clinically really understand the assays and how the data was generated and what it's good for and what it's not good for, because again, this is complicated. So we ran a very nice workshop as part of curating the clinical genome last year. It was in between the curating the clinical genome meeting and then the mutational scanning symposium, which is the big meeting for Atlas of Variant Effects. And so we had this really wonderful meeting where we got a bunch of people to either come early for mutational scanning or stay late from CCG to kind of get a bunch of experts talking to each other. We published a meeting report on this if you're curious what happened. There was, I guess the take-home message from there was a really broad consensus that it's important, and there was also a really broad consensus about how we ought to do it, which was very reassuring. And for someone who's running like a database project, it was really great for me because then at least, I knew what people wanted me to do, and I didn't have to try to integrate a bunch of dissenting opinions. Within Australia, we've been doing some needs assessments and workshops for clinicians and researchers to try to get them kind of up to speed on this, and we've also shared all of this stuff online. This is all freely available. I'll have a link at the end so you can see the slide decks that we've been using, and we're looking to build this out as an entire curriculum that we're going to also give at upcoming meetings, possibly in conjunction with like large society meetings, also online. And so we realized that it made a lot more sense to develop one set of curriculum that could then be customized to a local audience rather than have everybody make up their own slide deck. It seems like a lot of unnecessary duplication of effort, so we're trying to kind of coordinate all of that. So I'll just show you that it seems to be working. So here's some data from a workshop that we did in Australia. It was a very large workshop for Australia. We had, I think, over 100 people show up, which again, for Australia is very large. That's like everyone. Like everyone, all of the variant creation scientists showed up. So it was really fantastic. So, and that really demonstrated to us that we're doing something important because people bothered to come to learn about this stuff. And this question, which is maybe a little bit too small to read, is essentially just how confident are you on a scale of 0 to 5, and the top bars are before, and then after we can see this shift towards the higher numbers. I know nobody said 5, and this is cultural, so no Australians are going to mark themselves as a 5 for just about anything. Like even the people that we know are 5s and the people who are running the workshop would still put them at 4. I think you would get a different answer in the United States. But the important thing here is the shift. So we're really excited that this seems to be working. And and we got rid of all the zeros. So nobody went through the workshop and still thought that they didn't know anything. So that's good. Let's see, and this is a population of people where 90% of the attendees actually perform variant curation themselves. And so that's really the group that we're trying to reach. And so I'm very excited to talk to those of you at this meeting who are doing this kind of work, who may be interested in learning more and also thinking about how we can make sure that we're representing, when we're doing this kind of curriculum building and testing, that we're making sure that we're representing establishing oncogenicity and not just pathogenicity. And so with that, I hope that I have convinced you that high-throughput functional data can help solve variants of uncertain significance. And I would like to acknowledge a very large number of people who have been involved in this work. So I'm at the Walter and Wesley Hall Institute. We have support from the Australian Medical Research Future Fund, and that grant brings together people from all over Australia. We have a wonderful development team for MAVEDB that's split between Melbourne and also Seattle, where I have a lot of really long-time collaborators. At the University of Washington, especially Doug Fowler and Leah Starita. A lot of this work is being done in the context of the Atlas of Variant Effects Alliance, especially with this data curation and dissemination workstream, which gives us a lot of really good advice on what to do in sort of big picture. And then I've also really enjoyed working with collaborators within GA4GH to help develop the standards that we need and then also implement them to solve the problems that we have. And with that, I will give a little closing plug here for Atlas of Variant Effects. It's a grassroots organization. It's free to join. It's not just for people who are generating these data sets, but it's for anyone who wants to use them or get engaged. We have a clinical variant interpretation working group that is really specifically for people who want to use the data clinically and is trying to help develop guidelines. And then there's a link to the training workshop slides and other materials that I mentioned previously. And with that, I would love to take any questions that you may have.
Hi, Celestino. Thank you for your talk. One of the problems that I've noticed with a lot of functional data is that they don't take into account certain genes. So for example, Notch1 or CXCR4, the traditional loss of function type mutations are in one very specific area, right? And so what happens is, that some of these groups will think of loss of function across the board when it's actually not loss of function, it's gain of function that ends up being the cause because it loses the regulatory region or something like that. So how does these databases or that you're presenting take into effect positional changes that lead to oncogenicity in that case?
Sure, it's a good question, and I'm going to answer what I think is the bigger picture question, which is that we have data from a specific functional assay that tested some activity of the gene in some model, usually a cell model. And I think the challenge that you're talking about is sort of, I guess, the big challenge, the grand challenge for the field, which is how do you relate a measurement in an assay system to a phenotype in a patient? And sometimes that is very straightforward. It is often not straightforward at all. And I think that's something that requires a curator, an expert curator who hopefully has some clinical experience to evaluate the assay that was done and who understands the various presentations of disease that are caused by variants in that gene. And then can make a call and say well, this assay is informative for patients with these phenotypes and not with those phenotypes, or this assay will detect loss of function effects, but it will not detect gain of function. There's very few assays that can ever measure true gain of function. They can measure hyperactivity, but which is not gain of function. We can't. It's very difficult to measure true gain of function. So in systems where true gain of function is important, you would need to design a very specific assay that could capture that, or maybe do multiple assays. So I don't know if I really satisfactorily answered your question. The short answer is, we know that this is a problem and it's really hard, and it's not something that we're trying to automate because I don't think we can automate our way out of it. But what my view is, my role as someone who's running one of these data resources is to try to present structured data that provides enough context that would allow an expert to make that determination downstream. So.
Can I ask two questions?
Yes.
The first one is, first, this is wonderful work. I mean, I think this is just really the future, or part of the future, of variant impact classification. And so thank you for the talk. I was intrigued that you said you were able to reclassify some VUS and ClinVar. I'm wondering, when you did that, so basically you assumed that you were a well-established functional assay, right, or no?
Yes, and I think, and for those data sets, we were a well-established functional assay as per the Burnich guidelines on odds path, which was an independent assessment of well-establishedness, I guess, from the ClinGen SVI. So it's essentially a calculation that you can do that says how concordant are you with existing previous sort of independent variant classifications, and if you get everything right, then you can be strong evidence.
So it was a PS3 strong.
So BRCA1 is PS3 strong.
And then there was enough evidence for pathogenicity without that this kind of pushed it over the top, is what you're saying?
Yes.
That's fantastic. Really helpful. I guess the other thing, I was intrigued also that you said that a bunch of people are working on this really tricky problem of going from protein changes back to genomic changes, which is so difficult because it's not a one-to-one correspondence. Clearly there are many ways to get, from a change in the protein. There's many ways to get from a change in the genome to a single change in the protein, so it's easy to map that. But going back the other way seems so difficult, and I'm wondering if you have some insight into how you're doing it.
Yeah, so that is a huge challenge, and it's especially an interesting challenge when we have, for example, a cDNA-based assay that was performed in, like, say, a yeast model, because now it's been codon optimized to the point where there's not really a useful correspondence of the nucleotide sequence that was tested in the assay to the human sequence, although the amino acid sequence is the same. And we have many other data sets where scores were only calculated at the amino acid level. So in terms of how we approach that, we provide variants with respect to a RefSeq protein ID, and then it is up to the people that are using it to determine how they want to, whether they want to assign genomic coordinates to it. So for example, in the current representation that's in Ensemble VEP, they just went through and they enumerated all possible nucleotide changes that could achieve that single amino acid change, and then they just put those in there and they copied the amino acid consequence and copied the score over. I think that is certainly one way to do it. And it achieves the problem of saying if you have a nucleotide variant in hand.
The ClinGen allele registry also did that.
Right.
But I find that very confusing.
I find that challenging, and I don't want to do that for our data, because we do have some data that was calculated at the nucleotide level, and then we have other data sets that were calculated at the amino acid level, and I want to preserve that when we're exporting the data. I think that there are some new innovations in sort of genomic standards that are going to help us solve this in a more gracefully, and I look forward to adopting those when they're ready. But I think in the meantime, it's just, it's tough because, as you said, everything, expects, we want nucleotide positions for everything. We are also trying to, on that point, I will say, we're trying to be very careful to make sure that we flag assays where splice effects were not measured and some other sort of subtleties like that, because we wouldn't want to give somebody turn an amino acid variant into a nucleotide variant and then have someone assume that there isn't a splice effect when there may be one. And so there are some other sort of subtleties like that that we're working on. Should I just read this virtual question?
Yeah, so we also get Lynn a microphone, and then we do have a virtual question for you, Alan. What efforts are upcoming or underway to determine the criteria for the gold stars of MAVE functional data?
Yeah, so this is super hard because the data is so heterogeneous. There really isn't like a good kind of one size fits all. Like we can't, comparing to independent clinical evidence is very challenging because we don't have enough clinical variance for some of the data sets. It's very difficult to apply any sort of like generic statistical test to like establish goodness because the data is so heterogeneous, so many different systems. We don't want to end up unfairly penalizing a data set that was performed in an assay system that is very challenging to work with, for example, which would happen if we just tried to do something statistical. So we're thinking about this a lot. We would like to do, to be able to kind of anchor things based on something that exists for a lot of data sets, like, what's the separation between your nonsense and synonymous distribution is a reasonable proxy for quality, but of course not every data set includes that. So we're trying some different things and we're doing what we can. We will continue to be, MaveDB will continue to be a data warehouse and accept everybody's data, but then we're gonna try to be very, very discerning when it comes to what data sets do we export for clinical users.
Lynn, we have time for your question. That'll be it.
How many doctorates does it take to figure out how to use the microphone? So I'll just speak loud. So I'll try this. It's not the microphone. We need the. Okay. What do I do here? You just speak into it. Thank you. So So one of the things that I found a little surprising about one of the graphs that you showed very early, I guess the fact that the number of VUSs is doing this isn't surprising, but the number of known pathogenic variants seems to have leveled off. Can you comment on that? Do you think it's because the experimental science hasn't kept up, or because we need to be looking at things that, at regions that aren't protein coding genes, or?
Well, so that graph was only missense variants.
Ah, okay.
So it's definitely, so I can't speak to that aspect of it, of non-coding stuff. I think that, I mean, that it is going up, but I think the scale just kind of crushes everything down. But I think it's more a data integration issue, because we do have, for example, experimental functional data that could be used to reclassify a lot of these VUSs. But one of the things that we found through a lot of our outreach and stuff like that is that many variant curators are just not comfortable using functional data because it's difficult to spend the time to like go and read the paper and really understand the assay and whether it's suitable and so on. And the expert panels issue their recommendations, but they don't cover all of the genes. So I think that this is mostly a problem of, rather than needing to, while we certainly need to generate a lot more data, I think that we really just need to, in the first instance, we just need to do a lot more with the data sets that we have.
Thank you so much, Alan. We appreciate it.
Thank you, everyone.